So, good evening, everyone. Today will be a short lecture. Actually, we will discuss on the 9th day as per the workshop days. But as per the our workshops, it continued for the long time. Um, So this is bioinformatics, which gives an overview about this topic. So let's start. So we will discuss basically some biological concept in it, a brief history, where this bioinformatics data all came from, and some new words and techniques in it. So if we see basically your computational biology, bioinformatics, it's a quite interdisciplinary technique which has your biophysics, biology, mathematics, statistics, informatics. So they are all coming together in order to work for your in a good way. इसके अलावा you can see here that how the central dogma of life is playing a big role here. तो आपका ये डीएनए है, इसमें डीएनए जीनोम डाटाबेस है, देन आपका आरएनए है यहाँ पे, देन यहाँ पे कॉम्प्लीमेंट्री डीएनए ट्रांसक्रिप्टोम होता है, देन आपका प्रोटीन है, देन प्रोटीन सीक्वेंस एंड स्ट्रक्चर डाटाबेस के साथ मिला होता है, देन प्रोटियोम और फिनोटाइप ये आगे आपके आके प्रोड्यूस करते हैं. The National Center for Biotechnology Information (NCBI). According to NCBI, the basic definition of bioinformatics is that it is a field of science in which your biology, your computer science, your information technology, they all merge into a single discipline. आपकी biology है, आपका computer science है, आपका information technology है. ये तीनों merge हो जाते हैं एक single discipline में. So the ultimate goal of the field is to enable the discovery of new biological insights as well as to create a global perspective from which unifying principles in biology could be discerned. So there are three important sub-disciplines within bioinformatics. Three disciplines हैं, that is development of new algorithms and statistics, ठीक है, analysis and interpretations of various types of data, including your nucleotides, amino acid sequence, protein domains, and protein structures, and further development and implementations of tools that enable efficient access and management of different types of information. So tree of life has three domains. It's a bacteria, archaea, and eukarya. So the archaea includes your eukarya, eukarya, and bacteria. Yeah, and this is how other way of showing it. And there are two cells of types, as we know, that is your bacterial and eukaryotic. So bacterial includes your cytoplasm, nucleoid, cell wall, and cytoplasmic membrane, and the eukaryote, the respective bigger and better structure. The central dogma of your molecular biology, it includes on the left hand side is your prokaryotes, in the right hand side is your eukaryotes. So prokaryotes on the left hand side could shown that gene A and B, they are giving the transcription in order to produce mRNA. तो जीन ए से कोडिंग रीजन ए मिलता है, जीन बी से कोडिंग रीजन बी मिलता है, और जिससे आपकी ट्रांसलेशन होती है. पहले ट्रांसक्रिप्शन होती है, फिर ट्रांसलेशन होती है, और जिसके कारण आपका प्रोटीन ए और प्रोटीन बी बनता है. इसके अलावा यूकैरियोट्स में आपके न्यूक्लियस होता है, इसके अंदर एक्सॉन, इंट्रॉन, एक्सॉन, इंट्रॉन, एक्सॉन होते हैं, जो कि ट्रांसक्रिप्शन से आपका प्राइमरी आरएनए ट्रांसक्रिप्ट बनता है, एक्सॉन 1, इंट्रॉन 1, एक्सॉन 2, इंट्रॉन 2. देन आरएनए प्रोसेसिंग से आपके इंट्रॉन एक्साइज्ड होते हैं और मैच्योर आरएनए बनता है. देन ट्रांसपोर्ट टू द साइटोप्लाज्म, देन ट्रांसलेशन, देन आपका एंड में प्रोटीन प्रोड्यूस होता है. So you have alternatively spliced exons आते हैं आपके 300 नॉट 300 नॉट से आपसे सेपरेट होते हैं 600 नॉट से. तो आपका आरएनए जो फीचर एक्सट्रैक्शन है वो होती है, देन आपका प्रोडक्शन शुरू होती है इससे. फिर it's not required. This one is important here. So bioinformatics is mainly, it's all about genetically coded information and proteins. So DNA, RNA and protein ये आपके तीन हैं ये. और जो DNA है, DNA जो है रेप्लिकेशन से खुद प्रोड्यूस हो सकता है, तो ट्रांसक्रिप्शन से RNA प्रोड्यूस होता है, फिर ट्रांसलेशन से प्रोटीन प्रोड्यूस होता है. So it's all about data management at the end, like how much database structures you have, and how we do the analysis and interpretation of that data. We can use web-based or standalone tools writing code like Perl, Python, and Java. And beyond that, development of new algorithms and statistics is also important in it. Then, so whatever the things that we have discussed about the introduction part here, about the bioinformatics. So in summary, your bioinformatics, it helps to uncover biological information. जितने भी आपकी biological information is there in the mass of sequence, structures, literature, and other biological data, and obtain an insight into the fundamentals biology of cells and organisms. So it will include your genome, transcriptome, proteome, metabolome, and then at the end phenotype. So genomes to live computing. So on the left hand side is the computational tools and infrastructure. At the bottom is biological complexity. So Fraser et al. He produced this paper in the Science and shows these various way of biological complexity, having the molecule based cell simulation to be the highest, और आपका comparative genomics जो होता है वो lowest है आपका. Comparative genomics means to study genomes of various species in order to understand which genes are common with each other. You can also study genome scale protein threading, and beyond that molecular machine classical stimulation is also possible, and community metabolic regulatory signaling simulations. Then comes the data DNA. So raw DNA sequence informations, 4 bases AGCT, coding or not, and parse into the genes. The genetic code and the signals could be patterns in it. So if we are interested with this much of DNA for our bioinformatics experiments, so we will choose mainly this one. The rest is of no use. We will not concentrate our knowledge on that. And restriction enzymes are the sequence, as we know, they are the sequence specific. They recognize and bind to specific DNA sequences. And the enzyme E. coli, for example, binds to the 6 base pair sequence GAA-TTC. So it will cut from here. So we don't know how much E. coli could find this sequence within this sequence. So he found this one sequence, and this is possible mainly with your bioinformatics analysis. So you will put this sequence in there. So it will tell that after cutting from here and there, you will get your results. Further, as we know, there are purine bases and pyrimidine bases. Purine bases includes your adenine and guanine, whereas pyrimidine bases includes thymine and cysteine, cytosine. ठीक है, आपका नाइट्रोजन बेस होता है, थाइमिन होता है, गुआनिन होता है, एडेनाइन होता है, शुगर फॉस्फेट बैकबोन होता है. So all these things will play the role. Then base pairs may, within the DNA double helix, A forms 2 hydrogen bonds with T on the opposite strand and G forms 3 hydrogen bonds with C on the opposite strand. So like this. This is double bond and this is your triple bond. And this is a genetic code which consists of 64 triplets that we have discussed on the first class. So now the protein sequence, taxonomic distributions of sequence in UniProt or SwissProt. If we go there, you will see that 60% is the data from the bacteria, that is your prokaryotes, and the rest Archaea has 33%, and the eukaryotes has only 4%, and then 3% are viruses in the protein sequence. Then data it will have will be DNA, RNA, and protein structure. So this is the PDB molecule of the month. And on the left side is phenylalanine and aspartate tRNA. So some historical landmarks. 1953 structure of DNA was relieved, and in 55 the first protein sequence insulin, and in 60 first enzyme sequence will be there, and in 60s elucidations of genetic code. In 77 DNA sequence was done. Then in 75 first human genome was isolated, first human gene. And in 82 gene bank database is set up. In 83 PCR first describe was done. 86 fluorescence based DNA sequencing, and then genomics was coined, and the term bioinformatics appeared in scientific publications. In 1995 first complete genome sequence of free living organisms. In 1999 first chromosome was sequenced, and in 2001 your draft of human genome was found out. And in 3 it was human genome project completed. Total human genome was project was completed. Then comes your bioinformatics, which will include computational biology, then computational molecular biology, biomolecular informatics, and biocomputing. So in the bottom, what you can see is results by year on the left hand side, and the bottom is the years are going by 2010. So PubMed search with bioinformatics, so it started to increase from 2000 year, and the bioinformatics didn't see the back since then. It's keep on increasing like this. Then historical landmarks are like Needleman Wunsch first found out this logarithm for sequence comparisons. And in 78, Chow and Fastman prediction of the protein secondary structure from amino acid sequence. Then 81, Smith Waterman algorithm for sequence alignment. And then Kite and Doolittle hydropathy plots. Then 88, Pearson and Lipman search for sequence database. And in 87, progressive sequence alignment as prerequisite to correct phylogenetic trees. Then in 1990-1997, BLAST was done, PSI BLAST. So exponential growth in known DNA sequences are, so there are now more than 100 million DNA sequences stored in the gene database. So 1982, 1990, 2017. So all these with the base pairs are added in this sequence. And further, National Library of Medicine, it has 24 years of growth, NCBI data and user services. So like in 2004, you have PubChem started to increase its value, your base pairs. Then NIH, then genome-wide association studies, So the National Library of Medicine starts growing up and up and up and up like this. Then comes your DNA sequencing. So DNA sequencing with chain terminating inhibitors. So it will have high throughput sequencing like we have discussed in our third lecture, various kinds of DNA sequencing. At the moment, next generation sequencing are playing role, and in all these, bioinformatics playing a big role also. So first, in 1977, your organisms bacteriophage was sequenced. In 1995, your Haemophilus influenza from free living organism was found out. In 1996, first eukaryote sequence, Saccharomyces cerevisiae, was sequenced. So sequencing capabilities versus computational power. So if we see from the sequencing point of view, this blue one, it's from the 1996, it started to goes up till 10 raised to power 8, upar tak it goes. And in the green part, it goes like this, and by 10 raised to power 5, it goes still there, your computing part. So sequencing goes very high, whereas your computing goes bit low, millions instructions per seconds. This is the data showing you about the next generation sequence technologies that we are able to do with in 2005. We can do a lot of size per basis um, in order to find out the results. And this is the complete genome sequence available in the public domain, having more than 10,000 complete genomes. So, like Haemophilus influenzae, tuberculosis, Saccharomyces cerevisiae, C. elegans, B. melanogaster, human sapiens. Was draft was in 2000, and then yeah, like this. So in 2003, the Human Genome Project announces completion of the DNA reference sequence of human Homo sapiens. So chromosome 21, chromosome 22, they were like identified and they were published in Nature in 9th May of 2000 and 2nd December of 1999. This is the finished human genome project. Welcome Trust Sanger Institute. So these are the various sequences of the books that we have identified: 2, 3, 4, 5, 6, 7, 8, 9 till your 22 chromosomes, and X and Y. X has 1, 2, 3, 4, 5 books and Y has 2 books only. This is complete sequence of our human genome in this library. And then new methods and new words. So genomics, it refers to the analysis of complete DNA sequence, where the functional genomics, it understand they take the picture of genome functions, including the expressions profiles at the mRNA level and protein level. and the global approaches that are used to understand the functions of genes and proteins in it. Whereas the proteomics, which refers to the study of proteomes using technologies of large-scale protein separations and characterization. Whereas a transcriptome is a complement of mRNA transcribed from a cell genome. And the generation studies of complete mRNA expressions profiles transcriptomics. So further, structural genomics and proteomics. So it's include assignment of three-dimensional structures in order to find out the protein products and investigates the biological implications of these assignments. So this is the hypothetical structure, and its structural implications is paradoxin. So we can find in this way we can predict the structures whether it will be look like this or not. And omics also expands a lot with the genomics, transcriptomics, proteomics, metabolomics, pharmacogenomics, and physiomics. So genomics is the quantitative study of genes, and transcriptomics includes RNA and gene expression, as proteomics includes protein expression. And metabolomics includes the metabolites and metabolic networks. And pharmacogenomics includes quantitative study of genetic effects, host responses to drugs. And physiomics, physiological dynamics and functions of whole organisms. Then comes your transcriptomics. So this is your cell or tissue samples with cellular RNA in it. This is your microarray approach, and this is your RNA-seq approach. So microarray approach includes your label cDNA or RNA, and this is your probe in it. Then this infer for the transcript in it, analog quantification. And then on the other hand, it could go for polymerase enzyme in order to produce your cDNA or RNA with the help of RNA-seq approach, and then we can quantify that how much they are being produced. In the microarray approach, we have discussed in details about bacterial grown in culture labeled with complementary DNA with a Cy3 red dye, and other one from the infected person, Cy5 dye we took it. We mix them together, you have microarray, RNA binds to it, and you have these three different colors, green, red, and yellow. So we have just discussed various applications of our bioinformatics, how it could be used in different fields, and how from the past years, from 2000 year, it started to grow up eventually. In PubMed, if you put bioinformatics, it was not before. So after the Human Genome Project of 2003, things started to develop eventually. And in the RNA-seq, we isolate all mRNA and convert them into complementary RNA using reverse transcriptase. And then we sequence the complementary DNA afterwards. The map sequences to the genome at the end. This is your mRNA fragmentations, reverse transcription, sequencing, mapping, quantification. The more times a given sequence is detected, more abundantly transcribed it is. Then comes your proteomics, which is also part of your bioinformatics are used in this. So in the first case, you have your sample preparation. You take the proteome, you digest it, separate with the HPLC. Then you ionized with electrospray with MS full spectra. Then MS precursor selection. Then you do the quantification. Then do the fragmentation with the MS. Then you get the quantification of that data, including your identification and data analysis. Then proteome diversity we can see from one gene sequence to predicted amino acid sequence to many proteins. This is the whole complete gene here, and this is the predicted translated gene products. So this could be co and post translational modifications of the proteins and the end. For example, N or C terminal truncations, glycosylations, other modifications could be could be the role in it. And comes system biology. In order to understand our planet, it has ecosystem, then it has community, then has organisms, then system, organ, cell, DNA. So everything in this complex nature, we can create a human protein interaction also in order to study these two different part of our world. So in that also, your bioinformatics playing role. This is a way of understanding the bioinformatics data in which you have genome browser with extension. You can do various function with it like genes, markers, and microarray. And then you put your data, and then you get the gateway like various datas like this, which will give you data out with various protocol runner. This is like a typical bioinformatics way of doing it. So what are the top 10 challenges for bioinformatics? That is, it is a precise models where when transcriptions will occur in genome, initiations and termination, and it's a precise predictive models of alternative RNA splicing, and precise models of signal transaction pathways, determining the protein, DNA, RNA, protein recognition codes. Then accurate ab initio protein structures prediction, the rational design of small molecules inhibitors of proteins, mechanistic understanding of protein evolution, and their understanding of speciation, systematic ways to describe genes and protein functions, and development of bioinformatics curriculum. So that's it. It was not so big. So I will share this. So for today we are done. And rest we will do later. Okay, class. Take care. Bye bye. See you tomorrow. Same time, same place. Today we have a small class, not big.